So let's talk about Anaphase I of meiosis. Now, you know what? I'm going to go through the rest of the phases relatively quickly. So during Anaphase I, the microtubules shorten and the chiasmata are resolved. As a result, the homologs move apart from each other, one moving to one pole, the other homolog moving to the other pole. Homologs of each pair, that is. And you know what, I won't put another step there, but what I will point out is that the sister chromatids are still held together by the common centromere, because the cohesin between the sister chromatids that we talked about in the previous lecture, that cohesin at the centromere, it's still intact. It has not been degraded. So that's why those sisters still stay together. Now that's Anaphase I. We can see the homologs moving to opposite poles. Now let's take a look at Telophase I. So Telophase I, what happens? The chromosomes align, or arrive, I should say. The chromosomes arrive at each pole. The microtubules disassemble. A nuclear membrane forms around each group of chromosomes. And then in plants and animals I should say, most plants, most animals, cytokinesis ensues. So we actually get 2 new cells here. Okay, and those chromosomes might decondense also. So each of these products of meiosis I now will undergo meiosis II. So, okay, so what happens there during meiosis II? Now during prophase II, what does prophase II mean? Now prophase II means the prophase in meiosis II. So during prophase II, now this is very simple compared to prophase I. Essentially, if the chromosomes did decondense in telophase I, they will recondense. So the chromosomes condense, the nuclear membrane breaks down, microtubules attach to kinetochores. And you can see I have that diagrammed here. Metaphase II. What happens during metaphase II? Well, the one hallmark, the major hallmark, is that we will call these dyads. The dyads are aligned on the metaphase plate. And let me add the metaphase plate. Now I'm calling these dyads. Now anytime we see a chromosome that consists of 2 sister chromatids, now we can call that a dyad. I'm specifying dyad here because it helps us contrast the tetrads that are at the metaphase plate in metaphase I. So meiotic metaphase I, there are tetrads at the metaphase plate. Meiotic metaphase II, there are dyads aligned on the metaphase plate. So that's essentially it for metaphase II. How about anaphase II? So during anaphase II, that centromeric cohesin is finally degraded. so that centromeric cohesin is finally degraded, and that can split the centromeres, split the centromeric region of each dyad. So one monad now, one chromatid can go to one pole and the other chromatid can go to the other pole. So we have one going this way and one going that way, and that's for each pair. And I'm calling these monads here. So we can call any individual chromatid can be called a monad. So centromeric cohesin breaks down, the microtubules shorten, and that pulls the monads to either pole. And then that brings us finally to telophase II, which essentially is a lot like, telophase I. So the chromosomes arrive at the poles, microtubules disassemble, nuclear membrane forms, and cytokinesis, or cytokinesis, say it however you want. Cytokinesis ensues. Okay, so here we have, two meiotic products right here. Now, one thing I want to point out is let's take a look. Let's compare these, and I'm just pointing out that this is a monad here. Again, a monad is just another name for a chromatid. So monad equals chromatid. I don't know why we have all these different names that mean the same thing. So let's take a look at just one thing before we go on. Now, we started the hypothetical cell we used to go over the various phases of meiosis had, 4 chromosomes, right? 2 metacentric homologs and 2 acrocentric homologs. So we started with this over here, and we ended up with 2 of these. Well, remember that each of these right here can undergo meiosis too. Okay, so we can expect 2 more of these guys resulting from meiosis. And oh, my diagram is getting sloppy here. I'm kind of in a rush. My dogs have been in their crates all day. My little puppy has been, so I need to get home and let it out and take it for a walk. So okay, so what's going on here? These are the 4 possible meiotic products. We can count up the chromosomes here, right? 1. And how do we count chromosomes? We count them according to the number of centromeres. By convention, that's how we define, numbers of chromosomes in a cell. So we can see 1, 2, 3, 4 chromosomes over here. How many chromosomes in each one of these? We can count the centromeres. 1, 2. 1, 2. So same thing for these. Each one has 2. And what did we say? The main purpose of meiosis is to reduce the number of sets of chromosomes in half. So we have 4 chromosomes over here, 2 sets. And how many sets do each of these have? Each of these has 1 set. And a complete set includes 1 of the metacentric chromosomes and 1 of the acrocentric chromosomes. Okay, so meiosis did what it was supposed to do. We didn't get any combinations of, 2 metacentric chromosomes in the same product of meiosis. Well, okay, so that's meiosis.